minit lagi.
Okay, YouTube live now. Okay. Untuk presenter Dr. Husna ada prepare WhatsApp group juga. Ada apa-apa boleh bagi tahu dalam WhatsApp group lah. Sambil-sambil tu boleh jeling-jeling telefon. 5 minit lagi, 5 minit lagi, 5 minit lagi. Okey, oh dah live.
Dah live tapi dia
Okey, dia terus live kan. Tapi by default bukan from co-host kan, from host. By default check. Okey. Ni okey, sorry.
Assalamualaikum warahmatullahi wabarakatuh. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning. A warm welcome to all participants from Malaysia, the SEAMEO network, and affiliated countries to today's webinar. The theme today is Nutrition and Metabolism. We will be focusing on food and health. Our presenters today are from the Nutrition, Metabolism, and Cardiovascular Research Center, IMR-SEAMEO, at the Institute for Medical Research in Malaysia. I'm Dr. Nur Sufia Sulaiman from the same center, and I will be your moderator for today's session. Ladies and gentlemen, this webinar series was initiated with the aim to share the knowledge, discuss topics on biomedical research, and present research outcomes. Before we begin our presentation, allow me to announce a few housekeeping messages. We will not be taking any questions during the presentation, but participants can ask a question using the link that we shared in the email or in the YouTube channel. A Q&A session will be conducted after the three presentations. An attendance link is also provided for those who would like to receive an e-certificate of attendance. It is available at the YouTube description section. Ladies and gentlemen, for today's webinar, we have three speakers. Our first speaker is Dr. Fazliana Mansor, who is the head of the Endocrine and Metabolism Unit at NMCRC IMR. She holds a doctoral degree in biomedical sciences from the Karolinska Institute in Sweden. Her research areas of interest are biochemistry and molecular medicine in obesity, diabetes, and endocrine diseases. And she's also interested in the use of natural products in endocrinology. She has published many papers in international peer-reviewed journals. For today, Dr. Fazliana will be sharing her research experience in her presentation entitled A Lifestyle Intervention Study, Impact on Weight Change on Blood Pressure, Cardiometabolic Risk Factors, and Body Composition in Overweight and Obese Women. Dr. Fazliana.
Assalamualaikum and good morning. I'm Dr. Fazliana Mansor. Today I'm going to share with you on a topic, a lifestyle intervention study, impact of weight change on blood pressure, cardiometabolic risk markers and body composition in overweight and obese women. Another team member from IMR is Liana Ahmad Zamri. This is a collaboration project with the Institute for Public Health, NIH, or IKU, which was led by our late Dr. Safiza Muhammad Noor. Before we go further, let's look at the National Health and Morbidity Survey 2019. 1 in 2 adults in Malaysia were overweight or obese. 1 in 2 had abdominal obesity. On top of that, 3 in 10 have hypertension and 4 in 10 have raised total cholesterol level. We look at the earlier findings in HMS 2011, where the mean BMI of housewives was higher than other job categories. So that was actually why it was initiated. The project, this project is initiated. My Body is Fit and Fabulous at Home, or in short, my BFF at Home. It is a community-based weight loss intervention study targeting overweight and obese housewives in the urban areas in Malaysia. The study design is quasi-experimental, which was carried out around Klang Valley. The study population were among overweight and obese women who were not working or housewives. 14 PPRs were involved. The PPR is Project Perumahan Rakyat or People's Housing Project. We got the contact list from the State Health Department of Wilayah Persekutuan, Family Medicine Specialist, Community Clinic or previously known as One Malaysia Clinic, and community representatives. The map shows the location of the PPR, which they were divided into intervention and control groups. Okay, as mentioned, the PPR were divided into intervention and control group. It's not that the participants were randomly assigned into these two groups. And the study itself is divided, consists of two phases, which was the first 6 months is called the intervention phase, followed by the maintenance phase between 6 and 12 months. And the data was analyzed using intention to treat analysis. The assessments were divided, the assessments were done at baseline, 6 months and 12 months. I'm going to explain more on the intervention package later. So we measured the anthropometrics, body composition, blood pressure, and blood parameters. These are just among parameters that I'm going to discuss today. We have more actually that we measured for the whole study. The intervention group was given the moderate physical activity, individual diet counseling, and self-monitoring tools, where individual participant was given pedometer, besides 3-day food diary and physical diary that they have to record. As for the control group, it's unethical to just leave them unattended or not given anything. So they were given the general health seminars and also self-monitoring tools, but without the individual pedometers. For the cardiometabolic risk markers, we collected blood samples for the assessment at baseline 6 and 12 months. We measured the fasting plasma glucose, total cholesterol, HDL, LDL, triglycerides, and also insulin, which is important for the calculation of IMR. This is for the insulin analysis or insulin resistant index. We have these two machines in our lab for the analysis. For the analysis purpose, it is based for using the weight change category, which was described by Stevens et al. In year 2006, using these 4 categories. To our surprise, even the control group achieved maintained weight loss or loss between 2 and 5%. However, from the observation, more participants in the intervention group have lost weight or maintained their weight, but no significant difference were seen between these groups. For the cardiometabolic risk markers, we found group effect at the end of the study, which is at 12 months. This is significant, which is a good indicator for cholesterol. We also found reduction of fasting plasma glucose in the intervention group, which is also a good indicator. At 6 months, to our surprise again, we found a reduction or positive improvement in many of the parameters, even in the control group. So the first 3 is on the body composition, and they also have reduction in the systolic blood pressure. However, we couldn't see any significant changes during the maintenance or after the maintenance phase. We are quite happy with the improvement of the skeletal muscle mass, which was seen as significant in the intervention group compared to control. This is a good indicator that the exercise give a positive impact. So the decrease of visceral fat, which is the fat in your tummy area, in the abdominal area, and increment in skeletal muscle correlated with improvement in glucose and lipid metabolism. The improvement in control group could be due to factors such as attention, which is called Houghton effect. This can explain our observations in the control group, like why they also recorded or were seen to have positive changes, because they were probably aware of what they eat or what type of physical activities which they did which contribute to these positive results. So we saw all these positive results in the in the study groups. Of course, we have limitations where the quasi-experimental design may introduce bias. The chance of control contamination also may occur because the location of the intervention and control group within 30 kilometer radius, which the participants may have communicated among them and exchange information. However, we are also unable to generalize these results to other women population because this is according to the demographic analysis that we did. This is a low-income woman in the urban area. As for the strength, this is the first obesity intervention study in Malaysia that focused on low-income women in urban areas. The promising findings contribute to limited evidence in the local setting. So we conclude even from our baseline analysis, these women already have a significant or they already have the biomarkers that indicate cardiometabolic risk factors. The improvements in glucose metabolism, modest weight loss in lifestyle intervention benefits low-income overweight and obese women, and sustainability are possible. We have published a few papers, but the whole study is quite big, where we focus on the blood analysis, cardiometabolic, clinical and cardiometabolic risk factors. There are other papers that you can refer to which analyze other parameters. The photos show about health seminar, group exercise, blood taking, also how to analyze body composition using BIA or bioelectrical impedance analysis. I would like to express my appreciation to our late Dr. Safiza Muhammad Noor, who is very passionate and work hard for the study. The whole My BFF at Home team from IKU, Dr. Tahir Aris, who is actually the current director of IMR now, but who was a former director of IKU, who supported the project, Puan Rashidah Amba, and not forgetting our UPM collaborator, Associate Prof. Dr. Gita and Dr. Siti Yasmin. With that, I thank you.
Thank you, Dr. Fazliana, for the interesting talk. If there is any question that you would like to ask her about the presentation, kindly fill out the form using the link given in the YouTube description section. Next, I would like to introduce Dr. Aswir Abdul Rashed. He has been serving as the head of the nutrition unit at our center since 1998. He received his PhD in nutritional biochemistry from the University of Surrey in United Kingdom in 2012. His research interests are dietary intakes of vitamins and minerals in human cell lines, probiotic and prebiotic studies, development of Malaysian food composition databases, and development of intervention mapping for non-communicable diseases management program. He serves as a member of the Codex Committee on Methods of Analysis and Sampling, subcommittee member of Codex on Nutrition and Foods for Special Users. He is also a core team member in the Horizon Scanning Committee at the Ministry of Health. He is an EXCO member of the Malaysian Association of Research Scientists and is a member of the Nutrition Society of Malaysia. He has published many articles for academic and professional journals, book chapters, and conference papers on nutrition-related scientific studies. It is our great privilege to have Dr. Aswir with us to share his research. His presentation today is entitled, "The Potential Benefits of Edible Bird's Nest on Cognitive Function." Dr. Aswir.
Assalamualaikum and good morning to everyone. Thank you, Madam Moderator, Dr. Nosufia, for your kind introduction. Today, I would like to give a presentation entitled The Potential Benefits of Edible Bird's Nest on Cognitive Function. So, as an introduction, edible bird nest is made from saliva of the male shiflet, and there are two major species that produce EBN, namely Aerodremus fuciphagus and Aerodremus maximus. When we talk about its production, it comes from the cave or swiftlet farming, and this EBN is also referred to as the caviar of the East because of its premium price that can fetch up to $1,000 and can be considered as an esteemed food product in the East. The EBN consists of several glyconutrients, and one of the major glyconutrients is salicylic acid, which is around 9%. Glyconutrients are a group of sugars extracted from plants or food items that are thought to be essential for the body by helping cell-to-cell communication. And actually, there are 8 essential glyconutrients, and one of the most important glyconutrients is salicylic acid. This salicylic acid also can help to increase cell tissues repair, promote cell division, as well as cell proliferation. Okay, if you look at the role of the salicylic acid in the body, on the left-hand side, mostly in brain development and learning ability. In fact, at the early stage of life, salicylic acid plays a role in immune system and cognitive function. A study done by Z in 2018 presented the learning and memory function of the mice offspring when EBN was administered in maternal mice and during the lactation period. So the objective of our study was to determine the effect of salicylic acid from EBN on memory function. And for this study, we have conducted in vitro and in vivo work. So this slide is to show you the extraction process of salicylic acid, where we use the HPLC system to extract the salicylic acid from EBN. At the end of the presentation, I will share the link to get full access to how the extraction was conducted. We made the comparison at the same retention time, which is at 6.7 minutes for the standard salicylic acid as well as for the extraction. And then we collect the fraction and store for further use. Okay, once we have done the extraction, we conducted the salicylic acid uptake study. And before that, we have to check the confluence of the cell lines by using the TER. TER is a transepithelial resistant will be shared at the end of this presentation. So for the salicylic acid uptake study, at certain concentration of salicylic acid was added at the apical site of the chamber. So there is a cell monolayer with the permissible membrane that filter the amount of salicylic acid that can pass through the membrane to the basolateral site. And then we have collected this media as well as the cell monolayer and determine the level of salicylic acid by using HPLC. So once we have done the salicylic acid uptake study, then we follow with the Y-MIS test. Okay, and this Y-MIS test use BAP-C mice at 7 weeks old. It was conducted not to be harmful to the animal based on the in vitro study. The Y-MIS is a behavioral test for measuring the willingness of rodent to explore new environments. So we use 3 different concentration of salicylic acid plus the control group, and then we monitored and checked the mice for 21 days. Okay, this is the MTT cell proliferation assay. If you look over here, some concentrations show increased cell viability on all the selected cell lines. And then over here, 20 microgram per mil, 40 microgram per mil, as well as 60 microgram per mil show an increase in terms of the cell viability. However, a greater amount above 60 microgram per mil show reduced cell viability. And what does it mean? The concentration of salicylic acid that is too high is not recommended to be introduced or to be taken, because it may kill the cell lines. Okay, we use 4 different types of cell lines. KECO2 cells is originally derived from colon carcinoma, and one of its most advantageous properties is its ability to spontaneously differentiate into a monolayer of cells, and KECO2 cells being used quite extensively for that can mimic the gastrointestinal tract in our system. And the other cells, SKNMC, SH, SY5Y, as well as PC12, has been used extensively for Parkinson and Alzheimer study. So this is the result for the salicylic acid uptake study. Okay, the extraction show the highest level of salicylic acid at the basal medium as well as in the cell compared with the standard and the control. There was a significant difference between both extract and the standard. And what does it mean? Okay, this may be because of the purity of the extraction as well as the standard that been used for this study. And neuroblastoma cell lines show the highest amount of salicylic acid compared with KECO2 cells. However, we did not study the mechanism, but most probably because the presence of salicylic acid receptor and transporter is much more higher in the neuroblastoma cell lines than the intestinal cell lines. Okay, this is the result for the Y-MIS test. Okay, there was no difference in terms of food consumption and water intake in all the groups, and there was also no difference in body weight gain. However, if you look at the last table over here, you can see that the highest concentration, 60 microgram per mil, show the most number of entries to the novel arms and the longest spending time at the novel arms. So it means that the highest concentration shows some advantage and the cognitive performance on mice compared to the other arms. So what is the some of the conclusion from this study? The result of this study showed that salicylic acid significantly increased cell proliferation in neuroblastoma cell lines up to 60 microgram per mil concentration compared to the control. As if you remember the slide that I showed, it shows that okay, and then reduce the cell viability. And salicylic acid supplementation show promising improvement in brain cognitive function in mice. However, this study used normal mice, As I mentioned, it was BAP-C mice, and we did not conduct a study using knockout mice. If we did, I presume more interesting findings can be delivered. So before I end my presentation, I would like to thank COE Shiflet UPM grant for funding this study. Also to the Director General of Health and Director of IMR for giving their permission for me to deliver this talk. And not to forget to SEAMEO TRAPMET Malaysia for providing this webinar platform. Thank you. And these are the 2 published articles related to the project. And if any of the audience are interested to know more about this study, you can log on the website provided, the link provided. With that, I end my presentation. Thank you and Assalamualaikum.
Thank you, Dr. Aswir, for the interesting talk. If there is any question that you would like to ask him regarding his presentation, please fill out the form using the link given in the YouTube description section. Now, I would like to introduce Dr. Julaina Abdul Jalil. She's head of the Inborn Errors of Metabolism and Genetic Unit at our center. Dr. Julaina graduated as a medical doctor in 2003 at the University Science Malaysia. She is now a chemical pathologist who obtained her Master of Pathology in Chemical Pathology in 2011. She is also a visiting pathologist at the Biochemistry Unit at IMR. She is specifically involved in research and diagnostic of neurotransmitter disorders and lysosomal storage disease. Dr. Julaina will be sharing her research experience in her talk entitled Inherited Metabolic Disorder, or IMD, Bridging the Future. Dr. Julaina.
Assalamualaikum and a very good morning to all virtual audience for today. Firstly, I would like to express my gratitude to all of you for being able to spend some of your busy time to attend today's IMR-SEAMEO webinar series of year 2021. I will continue today's session with the last talk entitled Inherited Metabolic Disorders, Bridging the Future. Just briefly, I'm the head of unit of IM and Genetic Unit of Nutrition, Metabolism and Cardiovascular Research Center, NMCRC, Institute for Medical Research. Before I start, I would like to briefly summarize on today's talk. I would like to emphasize that my intention is nonetheless to create awareness to the audience on what is IMD. At least when looking at a patient with certain presentation, IMD is one of the causes that cross your mind. I would also like to share what are the researches conducted at our unit pertaining to this type of disorder. And I'm also honored to introduce our unit to the audience and also hoping to attract external collaborations in the near future. Okay, without further ado, I would like to begin my presentation. This is the outline of my presentation, consists of introduction, classification of IMD, current researches on IMD, briefly regarding our IM and genetic unit, and the last slide will be on acknowledgement. This slide is to show the normal physiology on metabolism of protein, carbohydrate, and fat in our body. Protein, carbohydrate, and fat, which we get from the food we consume, will have to go through specific metabolic pathways and biochemical reactions in order to produce energy, to synthesize new tissues, or to promote growth. So how does IMD occur? When there is a block in the normal metabolic pathway, IMD will occur. And basically, for a specific protein, for example, enzyme, receptor, transport vehicle, membrane pump, or structural element. This reaction, when an enzyme is deficient, substrate will not be able to be catalyzed to produce specific product, and substrate will start to accumulate in the body, causing harm to the patient directly, or go into alternative pathways, producing abnormal metabolites, which is toxic to our body. So you see, The principle for treatment of IMD is based on this pathophysiology of this disease. It could be by giving medication to reduce accumulated substrate, by diet modification, example by giving special milk, or removal of abnormal metabolites by specific medication. It could also be by supplying insufficient product or by giving enzyme replacement, which is the most expensive treatment for IMD patients, of about 1 million ringgit Malaysia a year for each patient. And to date, it only benefits patients with mucopolysaccharidosis. IMD affects anyone regardless of their ethnicity, their age, or gender. It is a new term and interchangeably used with IEM, or inborn error of metabolism. Okay, maybe some of you are more familiar with a portion of neonatal and childhood morbidity and mortality, which is about 1 in 3,000 to 1 in 5,000 live births. A study done by Dr. Zabeda and all back in 2008 showed that incidence in our population is not far from what has been reported from other regions. And this study involved 11 major hospitals all over Malaysia. Well, diagnosis for IEM or IMD is not only important for treatment, but also for genetic counseling for parents with child or children with this type of disorders and plan to have a subsequent child, or for the medical team to be prepared when they plan to have the next child. Okay, this next slide is to show how IMDs are categorized. There are few classifications of IMD. However, classifying them according to symptom is more practical and for me is easily understood. Type 1, silent disorder. Type 2, acute metabolic crisis. Type 3, defects in energy production. Type 4, organ enlargement and skeletal involvements. Type 5, neurological deterioration or regression, or other IMDs. Patient may be asymptomatic as in type 1, silent disorder, present as acute metabolic crisis, present with vomiting, lethargy, rapid breathing, or even sudden death as in type 2, or they can also present later as in more chronic disorders. Well, I hope by now everyone present today has already have an idea on what IMD is. Now we have a quick look into what are the current researches on IMD. Firstly, it is on developing potential biomarkers, particularly on mitochondrial and paroxysmal disorder. These disorders have wide genetic complexity as well as clinical presentation that they are difficult to diagnose. Therefore, developing biomarkers is crucial for them. And there are also studies on genomic, particularly using next gene sequencing or mass array to characterize IMD genetically. As for pharmacogenetic studies, it is more focused on LSD because as I have mentioned earlier, this is the most expensive type of treatment. And bioinformatics or pharmacogenomics are more advanced fields which are usually used for structural modeling, for development of new tests or study in treatment efficacy. There is also studies on improving performance of available assays by changing cutoffs in order to reduce false positives or false negative results. Besides that, there are also studies on development of second-tier testing, especially for confirmatory testing. Okay, now I would like to briefly introduce our team at IM and Genetic Unit with the strength of 2 medical officers, 9 research officers, 1 scientific officers, and 2 medical lab technologists. And what are the researchers pertaining to each officer? Okay, as for me, as a head unit, I'm doing research on neurological disorders. Dr. Salina doing research on glycan. Afandi Omar research on LSD, focusing on the treatment. Dr. Ernie Zuraida, research on bioinformatics and pharmacoinformatics. Fatima Diana, research on mitochondrial disorder. Balkis, research on sensors and biosensors. Nurjan Naim, research on mitochondrial disorder. And Dr. Praviti has recently joined us in earlier this month, and she has a PhD holder in molecular medicine. we have two medical lab technologists who are Siti Rahayu and Aisyah Falani, and I have 4 PhD candidates on the run. Muhammad Khairul Nizam doing PhD in Australia, Nora Sharina in UPM, Izian in UKM, and Rusnani in UM. So when they come back, I have more brains to do more research. Okay, there are type of diseases of IMD explored in our unit. The diseases chosen based on problem statement listed after we discuss with clinicians, as well as due to disease complexity, especially when there is involving diagnosis for those disease. Okay, as for neurotransmitter disorder, why we focus on this disease? Because we aim to form a national working group of neurotransmitter research, and to meet the criteria in order for us to join the international working group. Okay. Besides IMD, there are also researches done on other diseases. This is particularly because some of the officers are experts on certain equipments. Therefore, in order not to limit the expertise, I encourage on collaboration with other fields as well. Okay, so these are the list of researchers done on currently ongoing IM and genetic unit. In IM and genetic unit, the studies listed are involving our officers as a principal investigator. And there are many more researchers involving us as collaborators, which are not listed here. And as I mentioned before, the diseases are chosen based on problem statement in our population. Okay, for example, on potential pharmacological chaperone is done on MPS type 2 based on our publication, which showed MPS 2 as the commonest type of MPS in Malaysia. Okay, this slide is just to share what other equipment or technologies available at our unit. We really welcome to more collaborations in the future. As for now, we also have a quite number of collaborations with other MH hospitals and also universities. So we have LCMS, MS, we have GCMS, we have HPLC system, we have spectrophotometer for enzyme assay and ELISA. We have high performance computer, which is specifically used for bioinformatics and pharmacoinformatics. And we have automated DNA extraction system and a real-time PCR cycler for molecular work. We have a complete cell culture lab to do cell culture work. And we have one protein purification system, which is currently used for protein quantification. And we were looking forward to use this platform for other assays as well in the future. And we are going to hold the training on that in the near future. Okay, I think this is a slide where I would like to acknowledge these individuals for making this session possible. And I would like to thank all the audience for your kind attention. And if you have any questions, do not hesitate to email me at julaina.jalil@mh.gov.my. And if I'm not able to answer the question, I will channel the questions to appropriate officers. And I'll meet you in the live Q&A section. And thank you very much. Thank you again.
Thank you, Dr. Juliana. Now we'll be moving to the question and answer session. I have received a few questions from our audience. Let's just have a look. The first one is for Dr. Fazliana. One of our viewers want to know why the PPR, the People's Housing Project, why was it chosen for this intervention study?
Okay, thank you for the question. The target population for the study were housewives. So PPR has the right demographic for the study. And also the PPR has a, for each PPR, they have a community clinic nearby, which we could work together with. And on top of that, why we choose housewives and the demographic in PPR. We expect them to cook on daily basis and rarely eating out. So that would be a good baseline data, especially on the diet intake. I hope that answered the question.
Okay, thank you. The next question for Dr. Fazliana also. Do you study other aspects other than cardiometabolic parameters in the study?
Yes. As I mentioned before, this is actually a sub project. Sub data. The main investigator was from IKU, and there were so many teams. IMR only focuses on the clinical parameters analysis, especially the blood analysis. So there are other aspects that have been studied, such as even the readiness to take part in the intervention study, detailed diet intake analysis, and also physical activities.
Okay, thank you. Let's see. There's one for Dr. Aswir. What is the membrane pore size used for the sialic acid uptake study?
Okay, in our study, we use 3 micron pore size. Actually, there are varieties of pore size, but if we want to do any uptake study, so it has to be 3 micron or above pore size. Okay.
Thank you. So another one for Dr. Aswir. Is a piece of edible bird's nest enough to get the recommended concentration of sialic acid?
Oh, thank you for the question. Yes, actually, a piece of bird nest is more than enough because we use only 0.2 gram to get the cell acid, whereas the weight of a piece of a bird nest is around 3.5 gram. So it means that a piece of bird nest is more than enough to get the recommended cell acid for our daily intake. Thank you for the question.
Okay, thank you. Let me just check. There's a new question that just came in for Dr. Julaina. A patient with paroxysmal disorder, can they receive a vaccine, and which one is better for them? Oh, this is quite a tough one. We cannot hear you, Dr. Julaina. A speaker. Dr. Julaina, we cannot hear you. Is your microphone on? We'll just wait a while I think Dr. Julaina is trying to fix back her microphone. Maybe we'll just have a look whether we have any other questions. Dr. Julaina, are you on? Cannot seem to get through to her. Okay. Okay, maybe we'll just go on to another question that we have for Dr. Fazliana. Okay, the question is, was the diet of the women different from Malaysian women in general?
For Malaysian women in general, okay, in that context, actually, I think I have to look back at the detail of this nutrition data, because in general, not really different, but it depends on at what area or at what general diet that you're taking. Because in general, of course, our suggestion is now is suku spiral is the best one. Yeah, like quarter is the carbohydrate, quarter is the fiber, and quarter protein. But looking at today's diet, I think it's quite scary of what we are taking. That's why for any research, it's really important for the participants to really honest of what they are writing or what they are recording, because that is the only way, yeah, to get this even 1 week data or 3 days diary. So that's really important. I think for who's sending this question, I will get more data and maybe more precise answer for this one. Yeah, I might not be able to give that exact answer today.
Okay, thank you, Dr. Fazliana. Right, we're having a little bit of technical difficulty with Dr. Julaina's speakers, but I think we'll get back to your question through the email. I'm afraid that's all the time we have for questions for participants today. Any other questions will be answered by speakers to your emails. We hope that this webinar today has shed some light on some research that we have been conducting at our center and how health can actually be connected to the food that we eat and our metabolism pathways. We are also open to any research collaborations if anybody is interested. Ladies and gentlemen, we have come to the end of our webinar session. I would like to thank all participants for their support to make this session successful. Please give us a like on YouTube and leave any comments also, if you have any. You may subscribe to our NIH YouTube channel and click for notifications whenever there is a new post. Before I conclude, I would like to announce that there will be a for the next session of the IMR-SEAMEO webinar series. It will be on the 25th of June, so you can mark your calendars for that time. The theme for the next webinar is Dengue in Malaysia, an endemic amidst the COVID-19 pandemic, in conjunction with the ASEAN Dengue Day. We hope you can join us for the next session. Until then, many thanks from all of us at IMR. Take care and stay safe.
but I keep searching for your trace in the dark. Losing myself in the endless memories of the end right from the start. You're an obsession in my head, I can't forget your eyes. Your kiss was spread, my skin that feels so empty
To mislead, this is all you need. Be your everything, yeah, I'll be your everything. Still too soon to feel, please just say it's real. More than just a thrill, not just in it for the thrill. I'm in it for the ride, cause love at least to touch it. You're pressing on my body, babe, let me tell you something. I'm in it for the ride, cause love at least to touch it. You're pressing on my body, babe, let me tell you something. I'm in it for the ride. I'm in it for the ride. I'm in it for the ride. I'm in it for the ride.